The tracing technique described in this protocol helps us study neuronal morphogenesis and synaptic connectivity in the motor neuron system of Drosophila. The lipophilic dye can rapidly diffuse to every part of the cellular membrane with an extremely high density. This is why our protocol efficiently resolves the fine structures of a labeled neuron. If an axon terminal is accessible with an injection micropipette, this technique could be applied to any neuron in the larval and adult stages of flies or in other organisms.
If you have no experience dissecting or using a microinjector, precise operation on the specimen can be challenging. Understanding the anatomy of the embryo will help with guidance to proper positioning of tools to carry out dissection or dye injection.
To start, prepare and lay out all embryo collecting materials. Prepare the filtration apparatus by severing a 50 milliliter tube and cutting open a hole in the cap. Then set a mesh filter with 100 micrometer pores in between the tube and the cap. Prepare the dye injection micropipette and dissection needle from the same capillary tubing with an inner diameter of 1.2 millimeters. Pull the tubing with a micropipette puller at 7% from 170 volt maximum output to create a sharp needle with a taper of about 0.4 centimeters in length. To prepare the dye injection micropipette, soak the grinder with a wetting agent and place the needle on the micropipette clamp at 25 to 30 degrees, then lower the tip onto 2/3 of the radius out from the center of the beveling surface. Grind the needle while a syringe with tubing pushes air into it. Mark the micropipette with a fine-tip permanent marker to indicate the position of the opening at the tip after beveling, because it can be challenging to locate.